All right. Welcome back to Mr. Saucedo's YouTube videos. You're going to be drawing a diagram that looks something like this. So you might have to pause it just to get a diagram that looks similar. It doesn't have to be exactly this size, but it's going to be pretty good if it's as large as possible so that you can take notes and add and label things on there. But this is what I want you to draw. So we're going to be talking about what does the life cycle of a cell look like. So all cells go through different quote unquote stages, which we call the cell cycle. So generally speaking, a long time ago, it was divided into two stages because we had really crappy microscopes and observations were very limited at the time because of our poor microscopic images. So the first of those stages is called interphase. And so it is the large outside circle that's kind of this teal color. And so, yes, this is supposed to be the diagram that you drew, but I didn't want you to have to draw it three-dimensionally, so I kind of flattened it out. What is interphase? Interphase is when the cell appeared to be resting. And notice it is most of the cell cycle. So before we had good enough microscopes, people thought that cells were lazy and didn't really do anything. They're just kind of there. What's the other part though? So that other little yellow part that's on the outside of our main pie chart here is called the mitotic phase, or capital M. It's a phase when the cell was dividing. And notice that makes up a tiny little fraction of this. And that was when cool stuff was happening under the microscope. So they were like, oh, great, boring, 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 boring. Oh, something cool. And it's already done. And then it goes back to being boring again. And so that was kind of the view of what cells were doing pre-good microscopes. But now we have really good microscopes. So now we know that there are actually 5 distinct stages. So the first stage is actually G1. So this first little pie bit here that's kind of green, label that G1. What does G1 stand for though? It stands for gap 1. And that is when your cell is carrying out normal functions. So what's going on? The cell will slowly increase in size. Cell parts will start to increase in number. Because remember, eventually it's going to need to divide. So the last thing you want to do is be like, oh crap, we forgot to make an extra Golgi apparatus. Should probably do that. What happens at the end of gap 1 though? There is a restriction checkpoint at the end of gap 1. So before you can move on to the next little part of this pie chart, there is a restriction point. And that restriction point basically says, hey, is the cell big enough? And does it have enough cell parts to move on to the next stage? And so, again, we're looking at kind of like, this is kind of like going from infanthood to like toddlerhood, or toddlerhood to childhood, okay? Like it's going to be a change, and they want to make sure that everything's ready. Okay, great. Is everything prepared? Now, let's say that everything is prepared. Well, then we move on to S phase, or S stage. So that would be this kind of blue little chunky bit on our pie chart. That is DNA synthesis. So that is when the cell makes a copy of its DNA, because think about it, it's going to divide. You want to make sure that one cell has the complete set of genetic information, and the other cell that you are now dividing into is also going to have a complete set of genetic information. So that process is complicated, and we will go over it in Unit 8. So you don't need to know the details of it yet. But the idea is that you end up with 2 complete sets of genetic code by the time you're done. And you should have 2 complete sets of DNA if everything went according to plan at the end of S stage. Now, what's the next one? The next one is called G2. G2 stands for, you probably guessed it, gap 2. The cell is going to continue to carry out normal functions at this point. It will keep growing. It'll keep manufacturing, whatever it needs to manufacture in order to keep the cell alive. But at the end of gap 2, there is a critical checkpoint, and that will allow the cell to divide if it passes it. If it does not pass the critical checkpoint, so like if it is damaged, if there's not a complete set of DNA, if there aren't enough cell parts, if anything is sensed at this point that is out of normalcy, that means that the cell will basically kill itself. It's called programmed cell death. I thought of it as like order 66. It's literally just kind of like every cell in your body has this programming, And all it takes is the nucleus to say, okay, whoa, something's wrong. And it will tell the lysosomes to open up and to dissolve the cell completely. And so that's programmed cell death. Now, what is this little tiny sliver that's kind of like a light yellow? That's mitosis. And that's the process of the cell dividing. And at this point, the cell should contain two complete nuclei, right? So that's the plural of nucleus. But you should have two nuclei at this point at the end of mitosis. And so before the cell divides, it should have two complete sets of genetic information, and they should be contained inside of the nucleus of each of the nuclei should have that DNA inside of them. Now, the last step is cytokinesis. That is the process of dividing the cytoplasm, and you should now have two identical cells. In animal cells, the cytoplasm just pinches itself apart. So you can see that right here. It just kind of like we have two genetically identical cells, and they just kind of pop apart. In plant cells, though, remember they have a cell wall, and their membrane is not as flexible. So a cell plate will start to form. And here's a picture of a cell plate. You can see, actually, here's a better picture of the cell plate right here. You can see it starting to build basically a little cell wall to separate the two cells from each other. And once the cell plate has fused, now it is a cell wall. And keep in mind, though, that cells do divide at different rates. So the length of G1 is different depending on what type of cell is dividing. Why do you think that is the case? Why do you think that G1 can last, minutes, days, weeks, or years for different types of cells? Well, I'll show you in a little bit. But think on that. The rest of the cell cycle, though, so G1 can be forever. But S, G2, and M, that only really takes about like 12 hours. So that's the reason why when we had less powerful microscopes, they thought nothing was happening, because it's like, whoa, it looks like nothing is happening this entire time. And then all of a sudden, all this cool stuff happens really, really quickly. And so typically cell division rates from fastest to slowest are as follows. So let's see if you could have figured out why G1 takes longer than others. Skin cells. This whole process takes about 2 weeks. Red blood cells, 4 months. Liver cells, 300 to 500 days. Your intestinal lining, 4 to 5 days. Your actual intestines are replaced every 16 years. And this is not all of a sudden. It's not like you turn 16, all of a sudden, wow, your intestines are brand new. They're slowly being replaced all the time. Your skin cells are not all replaced every 2 weeks. Each skin cell is replaced every 2 weeks approximately, but not all of them are replaced at once, because you get a paper cut. Great, now that's new skin. That's going to start a new 2-week healing process. And so the reason why this is the case is because certain cells can last a lot longer than others. You don't need to replace your bones all the time. You don't need to replace your intestines all the time. There are certain things that you're going to need to get replaced over and over again, like your hair, your nails, your skin. But then other things can last a lot longer than that. And so do you think cell division would be faster in children or adults? What do you think? Answer? Children, right? Kind of obviously, it should be children. So here's the typical flow chart of fastest to slowest. So your stomach lining is replaced super quickly. Then skin, then blood cells, then liver, then bone, and then brain cells are on the far end, because most of the time they can't even be replaced. And so that's it. So if you have any questions, please let me know.